is a very brief overview of Mendelian genetics. Mendelian genetics is the foundation of contemporary genetics, though adapted for single-celled organisms that reproduce asexually. Some traits are traceable to the cooperative interactions of a number of genes rather than to a single gene. The twofold redundancy discovered by Mendel is due to two copies of each autosome, hence genomes of higher organisms are termed diploid. The two copies of a gene could convey different, possibly conflicting information. The different versions of a gene is called an allele. Organisms with two identical alleles of a gene are called homozygous, while an organism with two different alleles of a gene are called heterozygous. Genotype refers to the genetic or allelic makeup of an organism with regard to an observed trait, or in other words, is the sum total of sequence variations, including polymorphisms and mutations, that are present in a genome. Phenotype is the observed properties or outward appearance of a trait, for instance, height and eye color. When a gene is heterozygous, the observed phenotype encoded by one allele of a gene is dominant with respect to the phenotype encoded by another allele, the recessive one. There are different patterns of inheritance: autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, mitochondrial, and non-Mendelian, for example, imprinting. A well-known example of an X-linked recessive disorder is hemophilia, a blood clotting disorder. A person's biological sex is determined by the pairing of two sex chromosomes, X and Y, where males have an XY pairing with the X chromosome inherited from their mother and the Y chromosome inherited from their father, whereas females have an XX pairing. Women who are carriers of hemophilia have one X chromosome with one altered or causing hemophilia gene and one X chromosome with a properly functioning gene. One properly functioning gene usually provides sufficient clotting factor for women carriers to prevent excessive bleeding. So often women show no symptoms. Queen Victoria was a carrier, and you can see how the X-linked recessive trait was passed through the generations of the royal family to affect the males. In fact, Tsar Nicholas II of Russia had Rasputin act as the healer for his only son Alexei, who suffered from hemophilia. Historians believe that Rasputin used his position and influence to discredit the Tsaristic government and thus help precipitate the overthrow of the Romanov dynasty. Hence, it has been called the gene that launched a revolution. Other interesting examples of missense and other genetic mutations that lead to various disorders are described in this very readable book, When a Gene Makes You Smell Like a Fish. An allele that is present in the great majority of individuals within a species is termed wild type. That is, it is naturally present in large numbers of apparently healthy organisms. Though DNA is stable, the genome is corruptible. In other words, the genetic code can be changed. Mutations are when one allele is converted into another allele or an allele is created. The collection of alleles present in the genomes of all members of a species is called the gene pool for that species. Alterations in a DNA sequence can be inherited or acquired. Some common alterations include substitution, where one base is replaced by another. This could lead to either a missense mutation or a synonymous mutation, as pictured here. On the left, there is a missense mutation which leads to an altered protein product, hence a defective phenotype. On the right, there is a synonymous mutation, hence there is no change in the amino acid in the protein sequence. Therefore, there is no effect on the phenotype. Other alterations in a DNA sequence include deletion, where one or more bases are excised or cut out, and insertion, when one or more bases are added. We can see in this lower figure that the CD region from one strand was deleted and inserted into the homologous chromosome. That specific example is a duplication and deletion of homologous chromosomes. Other alterations in a DNA sequence include inversion, when a small subsequence of bases are removed and reinserted in the opposite direction. Somewhat related is a reciprocal translocation between non-homologous chromosomes, which is when a small sequence of bases are removed from one chromosome and then reinserted into a different chromosome. Since DNA is information, and information typically has a beginning point, such alterations in the DNA sequence would produce an inactive or altered protein. Transmission of a mutation from one generation to the next by the germ cells, that is, sperm and egg, is said to occur via the germline. Mutations affecting the genomes of cells everywhere else in the body, which constitute the soma, have no prospect of being transmitted to the offspring and are called somatic mutations. There are many chromosome disorders, though rare. For example, congenital abnormalities affect roughly 7 out of 1,000 newborns, though they are responsible for about half of all spontaneous first trimester pregnancy losses. There are also acquired abnormalities due to things like chemical exposures or behavioral choices, such as cigarette smoking, that lead to cancer. There are also single gene disorders. Individually, these are rare, but as a group, they affect around 2% of the population over their lifespan. There are also multifactorial or complex disorders, which are the result of a combination of genes and are thought to affect around 60% of the entire population. Again, if you are interested in genetic disorders, this book, When a Gene Makes You Smell Like a Fish, is quite interesting.